Okay, thank you for telling me. We're learning things here. It's always changing. All right, but you can hear me, right, Miley?
Yep, you're good. And I can see your screen, so.
Great. Okay, so I was saying that lab on Monday, you guys just need to bring yourselves pen, paper, pencil, whatever you want to write with. Otherwise, you're probably going to be good to go. Notebook, take notes, I guess. Whatever you would bring to a normal class, nothing too special. We're not doing anything particularly gross next Monday, so don't worry about that. And so that'll be up. And then the other thing that's important for me to say out loud to you guys is there's a Moodle page for 3314L, which is the lab for this class. I'm not going to be running 2 Moodle pages. All I'm going to do is put all the lab information below the lecture information on each week on the main Moodle page for 3314 that you guys already have. So you're going to have one that never looks like anything, and that's supposed to be. The lab information is also on the lecture Moodle. Okay, that's what was important, I think, for me to say right now. There's other things that I'll probably wait to say. And I'm glad we got this all sorted out, and we can go on. All right, so today we're kind of going into the applied world. We are not talking about theoretical stuff anymore. We're going to talk about some real phylogenetic trees, some real characters, some real synapomorphies, to give you guys context for vertebrates and how they fit onto the tree of life. I know that I have I always want to start really big, because I find that with a lot of classes that are an ology kind of class and ornithology classes and anatomy class, we're comparing vertebrates. It's always really easy to say, okay, let's talk about vertebrates. Starting with, and they show you like a full-on like bass or some fish, and that fish just exists out of nowhere, and you're going to compare that fish with an iguana and a cat or whatever. And I know I've made that point to you guys before, but I think it's really important, especially as biologists, and I know that from what you've told me about yourselves already, you have many different interests and many different backgrounds coming into biology. You're not all necessarily vertebrate animal focused, and that's like what you want your whole career to be. And so I think it's really fun to put us, because we are vertebrates and the rest of the vertebrates like us, into context with the rest of life. And so I always like to do something like this and just reinforce the absolute nightmare that is our universe, where like that little marble's got everything you've ever heard of on it, and then there's unknown amount of trillions of miles in every direction with nothing as far as we know. So that little marble's pretty awesome, and I like it a lot. Just a little context for vertebrate evolution. Okay. So the biggest units of life that we know of on this planet, the extant living forms of self-reproducing living things, are on Earth. And there's 3 domains that biology has recognized. These domains are way more recently understood and found and even put together than something like Linnaeus, right? These are from the last few decades. Some of these words are very old, like bacteria, and certainly biologists for like a century or so have known about eukaryotes. But things like archaea are single-celled life forms on this planet that, for all intents and purposes, sure do look a lot like bacteria. But because we can sequence their DNA, they're actually a completely different radiation of single-celled life. And so here's a phylogeny with no root. There's no point on this because this is everything. You can't have a root because this is all the stuff that exists right now on this planet. So where are animals? Go ahead and talk to your neighbor for a second. Vertebrates. Good. All right, and here in life, we'll say we're up in eukaryotes. You feeling good about that? We are up in eukaryotes, and right there, we are in this little clade of eukaryotes, and a monophyletic clade called opisthecons. You're not responsible for the word opisthecons. And to be clear, there's going to be a lot of stuff that I throw on these slides because I think it's cool for you to have access to it, but very little of it is going to be like, memorize this. It'll be clear what that is, I think, as we go. It's not about memorization. This is about exposing you to the bigger world. All right, so there's this big tree that's way too much. So let's simplify it. This is a phylogeny that I like because there's those groups, bacteria and archaea and eukarya. Eukarya has a ton of biodiversity in it. You guys are probably most familiar with the ones that are like visible with your naked eye, aka plants and fungi and vertebrates, animals like you. So there's eukarya. You can see there's relationships within eukarya, and then there's archaea and there's bacteria. What you're seeing on the far left is the history of the Earth. We know that this planet is just about 4.6 billion years old, and the oldest absolute for sure records we have of life are nice fossils from about 3.5 billion years ago, but there's chemical traces and hints of things in rocks that are older than that. And so what's exciting is that in 2023, our understanding of life on Earth is that for sure there's some living thing at 3.5 billion years ago. There might be living things as old as 3.9 billion years ago. These things, whatever they are, absolutely could be described as prokaryotic, and we're going to talk about what that is in a second, and I bet you know that word somewhere deep in your brain from intro bio or high school or something like that. What I think is cool is that this is a thing that scientists talk about. You guys might hear about this. LUCA is an acronym for the last universal common ancestor. So the thing that is a theoretical, what must have existed, population of organisms, population of self-reproducing blobs, really, that is the common ancestor of every single living thing on this planet right now. And so it's very, very fantastic that you and I, there's so much uncertainty around something like that, but that scientists from all over the world can have an extremely specific conversation about like where exactly LUCA might be, how old LUCA might be. Even if we don't know a lot, it's incredible that we have it constrained to there, I think. Okay. Those two groups of animals, bacteria, or animals, I shouldn't have said that. Those two clades of life, bacteria and archaea, those two domains, are usually set off against our domain. Our domain is eukaryotes, and these other ones are usually called prokaryotes. Talk to your neighbors for a second. I've circled the prokaryotes on this tree. How would you describe the clade prokaryotes phylogenetically? Chat with each other, make sure you agree.
Right, narrow use area. Yeah.
All right, so somebody tell me, as a clade, how would you describe prokaryotes? What does your group say? Yeah, Amy. A paraphyletic group, what does that mean again? Say a little more. So there's a common ancestor for all the things we're calling prokaryotes, but if we don't count eukarya, this is a paraphyletic clade of organisms. That's exactly right. So just like if we think of all the reptiles but don't count birds, that's a paraphyletic group. That's not very interesting for describing the evolutionary history of life, because I think you guys can see that things like archaea have a more recent common ancestor with us than either one of us do with bacteria. But obviously saying prokaryote has utility because these are all there's a few features, I won't say what they are, that unite those prokaryotes relative to eukaryotes that are, real. But those differences, and we're going to talk about them here in a minute, are more important and more interesting if you think of them as evolutionary derived features of eukaryotes rather than like two categories, have and have not. Anyway, we'll get there. So this is our first little voyage into using words like paraphyletic. So prokaryotes as a group is a paraphyletic clade, so not a great clade. We wouldn't really use it as a clade, but it's certainly an okay descriptor maybe of those organisms. All right, eukarya. Our domain of life is a monophyletic clade. You can see eukarya has a common ancestor, and all the descendants are within that clade called eukarya. I just can't resist telling you guys certain things like this. The oldest good evidence we have of fossil eukaryans is just under 2 billion years old, about 1.8 billion years ago. What is super, super, super fantastic about what humans know is not only can we find fossils that maybe are the earliest eukaryotes that are in rocks that are about that old. There's a couple that are actually a little older than 2 billion. But you can sequence the DNA of a bunch of eukaryotes that are alive right now and do models, random rates of mutation in the junk parts of their genomes that don't do anything and aren't selected for. You can sequence those pieces of DNA and then run an estimate on like when they would have diverged. And you get an estimate for the divergence of eukaryotes from living today DNA of like 1.9 billion years ago. And then the fact that somebody like me can go find a fossil that's 1.8, you can be like, lines of evidence letting us know that the evolution of eukaryotes, so the common ancestor of plants, animals, and fungi, is less than 2 billion probably. That's just really fantastic and really, really cool. You guys, I think, know what eukaryans are. So go ahead and take a quick second, talk to your neighbors. What are the features of eukaryotes? Intro bio trigger. All right, who remembers what are we dredging up? Some features of eukaryotes, usually used to set them off from the prokaryote. Yeah,
Organelles.
Organelles, what are organelles?
Although. That was good, membrane in case.
Okay. So there's organelles inside the cells, AKA membrane encased business centers, I guess. Stuff. Doing the taxes of the cell. Okay, so I'm not saying these are, I'm not gonna make this giant list here and say that 100% I agree with all of these going right there. This is the brainstorming. What else? Can people dredge up? Nucleus, thank you very much. That's the only one I have up there. So there is a nucleus, and what's inside that nucleus? Well, I'm just gonna even say the genetic material. Sure. There's a ton of features we could talk about there, but in a prokaryotic cell, there's like a ring of DNA that's kind of just in the cytoplasm as part of the hoop. In a eukaryotic cell, there's a nucleus, and that stuff's contained. That DNA might be all wrapped up around chromatid, the chromosomes, and very organized. That's totally possible. Not all eukaryotes are doing that exactly, but a nucleus with the genetic material inside. What other things? Yeah.
I think they're the only place that there's multicellular.
Oh, anybody know any multicellular bacteria? Interesting question. Philosophical question almost. Okay, so multicellularity. What's something that might go with multicellularity?
Oh, specialization of cells.
Oh, okay. So then you have like specialized cell. That bacteria's got to do it all, right? As one cell. You've got a nerve cell, a cell in your tummy, and a bone marrow cell. You got all kinds of stuff going on. What's something else that goes with multicellularity? Larger, bigger. Size, I like that. Did any other group talk about any other thing? There's one that usually goes with this one. It's not about the nucleus, but in my mind, I think it kind of goes there, that you guys all definitely know.
Multicellularity, different forms of reproduction.
That's true. There's different forms of reproduction. I'll get up here. All right, so you guys did a great job. There are features of eukaryotes. What I want you to realize is that some of those features in and of themselves are synapomorphies of eukarya, which means that like the presence of a nucleus in a dandelion and the presence of a nucleus in you is a homology way back. That's really cool. It's a synapomorphy of eukarya. So I would definitely summarize something like this. There's larger cells, there's more complex cells, there's DNA in a nucleus. They also have sexual reproduction. Bacteria exchange genetic information, but it's basically like chunks of their plasmids. A lot of eukaryotes recombine their DNA into new forms for reproductive purposes. So there's a little bit more for natural selection to act on in eukaryotes. Evolution can be a little bit maybe faster if you constrain all the other factors, because there's a little bit of mixing every time. So bacteria do exchange genetic information sometimes, but like actual recombinant sex is something that eukaryotes do. What I think is really cool about eukaryotes is that look how much time has passed since the origin of life, like almost we can think of it as close to 2 billion years of evolutionary time when eukaryotes have a shared lineage. And so you and that dandelion have so much of your cellular machinery, the actual details of your cell Yes, plants have a cell wall and you have a cell wall, but that's like one thing. Within there, how the Golgi apparatus works, all that kind of stuff. You and a plant are doing almost the exact same thing. So you might see in some newspaper or some scientific article, like, do you know that you share half your DNA with a plant? That's usually set next to a sentence about how you share like 98% with a chimpanzee. And that feels weird because 50% of the genes and something with a chimpanzee. But you should look here. This isn't like the base pairs, right? This is like functional genes that you share with like a banana or any plant you want to pick. You can pick pineapple if you want. That's really cool. And that has nothing to do with any of the features that make plants plants or you, but it's the features that make eukaryotes eukaryotes. This is the one I wanted you guys to say. So mitochondria. Yes, you did. But there's all kinds of organelles, right? There's like whatever vacuoles, and vacuoles are just big warehouses. A mitochondria is something really specific and really intense. So what is a mitochondrion? Chondrium? It's a bacteria? What do you mean by that?
First is bacteria, or I can still have bacteria, and it's been known.
Endosymbiosis? Do you remember that? Yeah, go ahead, sorry, Will.
It's basically works now inside cell.
Inside a larger cell?
Yeah.
So here's a good like diagram, right? Here's just like a eukaryotic cell cartoon. All these red things are supposed to represent mitochondria. And mitochondria are ancestrally their own bacteria. They're their own thing. And now they live forever inside of our cells. They reproduce inside of our own cells. When your cell divides, the mitochondria all do a bacterial divide. And then half of them go with the new cell and half of them stay with the mother cell. So there's size difference. There's a bacteria living inside a eukaryotic cell, or things we can call an ancestrally bacteria thing. Is it really its own thing anymore? No, mitochondria can't live outside of eukaryotic cells anymore. They make energy. None of you said powerhouse of the cell, but that's what they are, right? This is where ATP is made, is inside a mitochondrion. And so all of this complexity and size and specialization is powered, and part is powered by a symbiotic event. This is still a single-celled organism, but it's got a bunch of other single-celled organisms effectively living inside of it, and they're dependent on it, and they make all the energy it uses. That's endosymbiosis. These things are symbiotic with each other. This bigger single cell, and then all the little bacterial things that live inside of it. So a plant and a mushroom and an animal all have mitochondria. Mitochondria is, like I think, probably one of the most important possible synapomorphies of eukarya. That is very, very cool and very freaky. Okay, some people have done a lot more work on archaeans. We didn't know about archaea like 30 years ago. So the study of archaea is still happening, this clade of organisms. This is how some of archaea falls out now. The group archaea is also paraphyletic sometimes relative to archaea. So that's just showing you guys, I want you to take this in not as anything important to remember forever, but just like science is ongoing. And so there is like a core group of organisms that are like the original archaeans that were ever discovered. But some of the things that we call archaea, we now find as like non-eukaryan things that are on the line to archaea eukarya more than they are to archaean. So the point I'm just trying to make is that biology is ever evolving and complex. I think that's really cool. What's another kind of endosymbiotic thing going on in eukaryan organisms, or at least some of them? Yeah, chloroplast. What's a chloroplast? The light capturing organelle, right? Photosynthesis happens inside of a chloroplast. A plant, like a dandelion, in its leaf cells, has mitochondria. It also has chloroplast. The chloroplast are where the chlorophyll is. That's what makes the leaf green. But what is a chloroplast besides being a sun factory to make sugar? It also used to be a bacterium. So there's multiple points on the tree of life where bacteria become endosymbiotically enmeshed with eukaryotic or eukaryotic organisms. That's crazy. And this evolutionary tree helps us see how that works. So plants have mitochondria, and that presence is homologous with your mitochondria, but they also, again on their own, did another one. And they have chloroplasts that do a completely different thing inside their cells, which is really, really cool. This is this phylogeny of life that I absolutely love. It is not aligned with this timeline over here, but I like for you guys to look at it. I just think it's incredible. When you think of like, where does oxygen come from in our atmosphere? You always say things like plants. So here's plants and algae. They produce oxygen. They take in carbon dioxide and they let out oxygen. But actually, cyanobacteria, which is a lineage of bacteria, are the only organisms that have ever evolved photosynthesis on their own. And they still do, as blooms, produce oxygen. But in fact, there's a lineage of bacteria that got enfolded into the ancestral eukaryote way back here. And there's a lineage of cyanobacteria that got put into cells of plants and algae later in time. And again, just being able to like talk to Darwin or talk to somebody from the 1920s and be like, do you know what we really know now in detail is like the timing and the phylogenetic situation of these endosymbiotic events. It's not just like fungi have mitochondria, but they grow like plants, but they don't have chloroplasts. What do? We know so much more than that now. And I think it's cool for you guys to take in that there's like these endosymbiotic events that help us get to the complexity of today. Any questions or anything to say about that? Super not vertebrate comparative anatomy, right?
Mitochondria came in to eukaryote twice separately. Is that a dispute?
I'm not sure about that. I don't know. I bet it's possible there's multiple events, but I don't know. Here is a nice, I think, phylogeny of eukaryotes. This is actually like zoomed in. A bunch of the clades of eukaryotes are visible here. So I can mark on the tree, here's the monophyletic clade, the node eukarya that contains a common ancestor and all the descendants of that common ancestor. That's eukarya. You guys are going to see trees like this all throughout this class. Mitochondria we can make as a tick, the presence of mitochondria here as a synapomorphy, a derived shared feature that unites all those eukaryotes. Chloroplasts, I put a little asterisk that I'm not going to talk about because this isn't the right class to do that, happen here and lead to these organisms, but not all the rest of them because it only happened on this lineage. Do we recognize things here? What if I do that? Is that helpful? There's a couple more clades we can talk about of eukaryotes. So there's plants over here. There's animals over here. Do we recognize anything else amongst eukaryotes? Oh, I heard a great word. Whose brain is blinking with this word? Who knows what this word is or remembers what this word is supposed to be? If you don't, it's fine. We'll forget it for now.
Basically, the other cube that explains the other.
So you guys might look in a biology textbook, you might have had in high school biology or something, people say like bacteria, protista, plants, fungus, animals. And what protista always was all the single-celled eukaryotes. You're a plant, you're a mushroom, you're an animal, or you're some kind of eukaryote. And so all these things, and this one, that's an amoeba up there, we're always called protists. That's a nonsense clade because now we have DNA and we actually have like a tree of life for eukaryotes. So we can talk about things. Please talk to each other about this, take it in, let me know what you think is going on. Just chat with each other.
Yeah.
Spell that.
All right, what are some observations or conversations that are happening right now? Not looking for anything in particular. Yeah.
Multicellularity was involved in everything.
Oh my gosh, this is a tree that you could be like, hey, I need to see the data for this, and we of course could show you. But yes, this tree is showing you multicellularity evolved a whole bunch of times. So is this something that could characterize eukaryotes? Absolutely. You are living proof you are one. But it's not as big more people clay. Most eukaryote diversity, and of course, all the time when all these endosymbiotic events happen, they're just big single cells too. And there's a ton of diversity of single-celled eukaryotes today. So multicellularity is not a feature of all of them. It's happened again and again and again and again and again. That is cool and very interesting. What other things were we talking about?
I said that it must be pretty nice to be multicellular, considering that it has shown up so many different times independently. It must be advantageous.
Can we all imagine universes where in the puddle you're in, at the ocean shore you're in, or whatever it is, being multicellular could be selected for? I think so. I think that's totally valid. There's obviously nothing wrong with being single cell because those bacteria are just like, they've been there the whole time. But multicellularity can be something that gets selected for, and it has many, many times. What other things? Just so you guys know, the basic fact of the multiple originations of multicellularity is usually something that I'm like happy that you guys are like, oh wow, because like I didn't know that. I didn't think about it like that. Especially when I give you things like this. You know, multicellular, multicellular, multicellular. You think, okay, eukaryotes are multicellular. Those 3 times are all their own time. Fungi did it, animals did it, plants did it. Who else did it? This little thing here. This is called, you guys might hear about this, especially if you ever go to the ocean, red algae. What's that thing over there? Green algae is here, part of the plant group. Plants are a fancy pants kind of green algae. Palm tree algae. I bet you know a word for palm tree algae. That is kelp. Doesn't it make you want to scream that kelp is so not close to plants?
Doesn't kelp also have chloroplasts?
It does. That's why there's a stupid asterisk here, because I didn't want to bother explaining it to you guys. But there's multiple times of endosymbiotic events with chloroplast-like things. It's actually probably lateral gene transfer from things that you would call green algae, with like endosymbiotic events coming over from plants. It's a nightmare, and this is not that class. But yes, these photosynthetic, they're all their own thing though, relative to plants, but their chloroplasts kind of go back to the plant chloroplasts. It's a nightmare. And again, not this class. But next time you guys are on a beach, if you ever find yourself in California or something and you see a big piece of kelp, pick it up. Because like the leaf, which functions like a big leaf, does not have any of the anatomy in another leaf. It looks like a sheet of like plastic. It looks like cellophane. It's its own thing. It's crazy. Anyway, that's all. No more brown algae. We're done. I wanted you guys to have this though, because I think it's really cool. Multicellularity is involved a bunch of times in green algae, which includes today's plants. Red algae, kelps, fungi, animals, and tons of eukaryotes sometimes become multicellular just for a purpose. Anybody know about the slime mold thing? Slime molds are single-celled decomposer kind of things, but sometimes like 100 of them will become what looks like a slug and work together to like go somewhere and then reproduce. I'll let you look that up on your own because it's not compared to vertebrate anatomy. So all of this crazy evolution, all of this time is taking place on the back of geological history. I'm not going to go into it. You're going to hear me use words from it before. I'm just giving you this slide so you can go back and look at it. There's nice descriptors of like the different periods of Earth history and important things that happened in each one, if it's of your interest. What I actually just think is important for you guys to understand is the scale of time that happens on Earth with all this eukaryotic stuff relative or prokaryotic stuff, relative to all the vertebrate stuff we're going to talk about for the whole rest of the semester. So there's a ton of ways that scientists can like take the 4.6 billion year history of the Earth and like make an analogy. What if it's a football field? What if it's the distance to something in your town? What if it's a year? I like the year one. So if Earth forms at midnight on January 1st, you don't get life at all until late February. And then here's all these other things that happen. Importantly, vertebrates don't show up until November 20th of that year, if you take Earth history to the year. So it's almost Thanksgiving before vertebrates show up at all. And then there's a bunch of other fun stuff. There's not really stuff on land. Land plants on the 22nd. There's not vertebrates on land until December. So there's no vertebrate walking around until the last month of the year. That's how much time is happening in this single cell stage, why you have 50% of your genes with a banana, because all that evolution is happening in that huge amount of time. This is, of course, the funny one. We don't show up until 24 minutes to midnight, and we don't start building cities and growing crops on purpose until people are going 58, 57. And that's just a little scale for you. Go ahead and enjoy that at your leisure. Just a little bit more context for animal evolution, because that's what I actually want to talk about today is vertebrates and animals, now that we're well within eukaryotes. So you guys are going to see these here on the side of some of the slides. So I also teach paleontology, so I already have these slides. You are not responsible for any of this in terms of knowing it. You do not have to know it. I just have it here, so you have it as a reference. And so this period of Earth history, the Phanerozoic eon, that's what we're in right now. That is the area of visible life. Almost all good fossils come from this blue bar, which is about the last 550 or so million years. That is when animal evolution really goes buck wild. The oldest fossils we have of truly things that we know to be animals come from this period called the Ediacaran period. That's about here. There's when it is, 635 to 541 million years ago. We're not going to spend too much time talking about it here. This is an example of like an artistic restoration of what some kind of life looks like. Everything's smushy, everything's jelly. There's not a lot of hard parts in the Ediacaran. There's obviously cool things going on. These giant pillars back here are big bacterial mounds. There's photosynthetic bacteria, cyanobacteria. They're building mounds and growing like coral does today. It's really cool. What's interesting is that some of the single cell eukaryotes that are sister to animals, they look like this. It's a little cup with a flagellum that sticks out of it. It doesn't move, it spins the flagellum, it draws water into the sides of itself, filters food, and then the water goes out. What's something that biologists recognized hundreds of years ago and were like, this is weird, is that these animals today that are single cell, that's what the single cell looks like. And if you take a sponge, which is one of the most primitive kinds of animal that's alive right now, and you cut up a sponge into its smallest part, this is what individual sponge cells look like, which is pretty much like if you glued a bunch of the sister group animal things that are totally still alive together, you'd make a sponge. Sponges are animals that all they do is they have thousands of little tails inside their bodies, and that draws water into the sponge, and that water goes out the top. Go ahead and Google a video of like a sea turtle laying down on top of the sponge, and there's like a little jet coming out. It's really nice. But the sponge is a filter feeder, multicellular animal. I shouldn't say that because it is absolutely an animal. And then the outgroups, the protists, the single cell eukaryotes closest to animals basically look like sponge pieces. That's really cool, really interesting. I'm only showing this to you guys to give you like some of that evidence we have for this ancient diversity that we know of for animals. These are two of the best known kinds of animal from the Ediacaran. We don't really know that much about them. They're basically smooshes in the rock. What we can tell about them is that they have symmetry down the middle. They're bilaterally symmetrical. We're going to talk about what that means here in a second. I just think it's cool for you to see that that's as far back as it goes. So that worm and that sponge emoji are supposed to be showing you on the timeline about how far back we have good animal fossils. Here's that geological time scale again. This is now after the Ediacaran. This is all the different words scientists use to cut up Earth history to the present. So you and me are sitting right here in the modern day. T-Rex lives back here in the Cretaceous. Stegosaurus lives back here in the Jurassic. I'm not talking about this today. I'm putting it on the slides so you guys have it. And when we talk about things later, it'll be something you've seen before. But I'm not going to focus on it today. Now is about vertebrates and their origins and also where they fit with animals. So once we get into a monophyletic clade called Animalia, which themselves, of course, are also eukaryotes. Animalia has 36, maybe 37, maybe 38 phyla. So a phylum of animals. This is a subsample. It's not quite that many, but it's a lot of the better known ones because the ones that are questionable are like real nightmares. Here's a bunch of animal phyla. Each one of them monophyletic, each one of them their own thing, each one of them quite ancient. Please talk with your neighbors and tell me about animal diversity at this level. Go ahead. And I chose a lot of pink and purple on purpose. That's not a thing. Okay.
Please.
Okay, I am very much enjoying what I'm hearing. I'm gonna ask you to do a little more too. We're not gonna go get lost in this, but I'm just curious. What are the phyla up there? And obviously you might just recognize the organism, but you're like, I know that one, I know that one. Just curious. Go ahead and talk to each other.
I know.
All right, so the first thing I asked you guys about, just observations you can make about animal diversity. What are some observations you guys have as groups? What are your groups talking about when it comes to animal diversity?
There's a whole lot of worms.
A whole lot of worms. Tell me about what is a worm? Long body, what else? Smushy, soft, smushy. Anything else?
But there's different kinds of worms. Worm is just a blanket statement.
Of course it is.
So
When you guys say worm, which one of these do you mean?
there's an earth.
Here's the one here's the one that if we went and we're very mean, we could go dig up right now because they are definitely not trying to be awake right now. It's too cold. But we can go outside right now and find one of these right here. This is a phylum of animals, earthworms. They're called annelids. There's also ocean worms that are annelids. So annelids is one phyla of things that we might be like, that's a worm. But you guys said wormy for a lot of other stuff too, and I'm not going to say you're wrong about the gestalt of a worm. So worms are long, smushy. What else? What'd you say? No limbs. Ooh, bilaterally symmetrical. If you cut them in half, you do it like this. Again, a mean thing to do, but maybe you're fishing and cut them in half like this, maybe. Are all animals bilaterally symmetrical? You can cut them in half like this. Interesting. What other features, what other observations are there?
Do they need to have a distinguished head and tail?
A distinguished head and tail? Who does they? What are you talking about?
Is that a feature of all worms or just certain planes of the worm?
Ooh, I'm not gonna say, so the worm, lowercase w wormy body plan certainly has a front end and a back end. And that front end has all the sensory structures, usually like the antennae and the eyes or whatever. The mouth is usually up in the front. We're going to talk about that in a wormy body plan. What other observations people have though besides it looks like a lot of worms?
These guys aren't closely related.
I mean, they're all animals. So, well, no, I'm not saying you're wrong, but like from this picture, what do you mean?
Like, I mean, we've got a flamingo, and we've got a spider, we've got but those are all very.
The flamingo, the spider, and the octopus are for sure the hashtag celebrities of this picture, because most of the other ones are like what? Like maybe the jellyfish, I guess, or the sea urchin. Okay, but you're saying they're not closely related, but at the level we're talking about right now, before we keep zooming in, they are all animals. So there is a lot of definable features of animals that we're going to talk about, a couple of them.
There's still like a lot of biodiversity that each of these pictures are.
Oh, yeah, there's a ton. A great white shark is represented by this flamingo on this image, right? This represents all of the backbone animals, and I just picked the flamingo because it's a funny choice. And why not? Tell me why not? You can't. So I picked a flamingo for fun.
I think worms are like the protists of animals, where they're just kind of killing a bunch of.
Look at this. The protists of animals, the prokaryotes of life. Interesting, interesting. All right, what else? What's the habitat? Thank you. What kind of water? Ocean.
Some of them are parasitic.
Some of them absolutely are parasitic or have parasitic members, no question. Something I do want you guys to take from this though is the simple truth that animal diversity at this high level, so the phylum level, is absolutely dominated by marine organisms. Most animal life is at this kind of a scale, the phylum scale, is marine and has never not been marine. There's only a handful, and guess what? They're the popular ones, of animals that have gone into freshwater and animals that have gone up onto land probably after that. Mollusks have done it. You guys know snails and slugs and octopuses. Arthropods have done it. Hashtag bugs. Vertebrates have done it. But most animal phyla are marine organisms only. And so if you have a tree of animals and you reconstruct all of where they come from, you're like, oh, wow. I bet you the common ancestor of all these living things was for sure in the oceans. And then you can go to the museum and be like, hey, fossil people. And they're like, they only come from the oceans. And you get to be like, I just learned something about animal origins, marine. That is super, super cool and important, I think. So most of these things we're not going to talk about at all because they are just kind of too much diversity almost. But again, as biologists, I feel like you guys need to be aware of this. Any last thoughts on this before we go? There's a lot of fun things here that I know we have to leave, but just curious.
I mentioned that all of non-worm ones have worm members. Except spiders, which are hard.
Spiders are hardworms?
Sponges are.
Oh, sponges are hardworms.
Spiders have.
I don't like that. I don't agree with it. Okay, so now cutting off a lot of the chaff, cutting off a lot of the like crazy phyla that you guys are going to never have to know anything about. Here's more of the celebrity phyla all put together. This is the phylogeny. This is how the phylogeny works. This is definitely rock solid. Cartoon versions. Here's some words for the animal tree of life. That one's easy. You are responsible for that one. Animalia is a monophyletic clade. There's everything that's inside of it. Within Animalia, there's a lot of good diversity that we have names for. This clade is called a bilateria. I didn't say this yet, but I should have. On all the previous slides and this slide, anything with a phylogeny, if the word is fully capitalized, vertebrates, which is what this class is actually all about, are a member of that clade. So vertebrates are bilaterians, they're animals, they're eukaryotes. Okay. As we go forward, that's important because you're going to see a lot of names, and if it's not all capitals, it's not about vertebrates. But it just happened. So here's bilateria. This is a monophyletic clade. That thing, and the damselfly, which I guess I should have kept a spider, he is particularly not close to the flamingo, but they both are bilaterians and they both are animals. Within bilateria, there's a split that we're going to talk about. Deuterostomes and protostomes are two monophyletic clades, sisters within bilateria. Most animal life is protostome. Most animal species are protostome bilaterian animals. There's a couple clades there that I'm putting, you don't have to know them, but a Dysozoa, Lophotrochozoa, that's just a really fun one to say. Say it to your neighbor, Lophotrochozoa. You're not responsible for it in any way, I'm just showing you where it is because it's important. All right. What I think, I'm gonna show you that in a second. Well, I'll show it to you now, because I think whenever I talk about animal diversity, I just can't help it. This is what I think of the whole time, is because that show has a very good biodiversity. Just to be clear, very good, totally accurate, and there's only 2 vertebrates, right? They're both women. Interesting. Anyway, so is the sperm whale. Anyway, okay, fine. Talk to each other. Look at this. You guys know how to read phylogenies now. Follow back to common ancestor. Farther back. Just get a sense of it. Here's a phylogeny now, not a bunch of random pictures. A phylogeny of animal diversity. What are some things you notice? What's interesting? I'm not looking for a pattern. I'm just looking for you to take this in and like talk about it for a second. So talk to your neighbor. What are you noticing on this tree? What do you have a question about? Maybe go ahead and take a second.
The octopus is like.
Evolution, explaining this more. Like. That was.
All right, what are some observations you have, questions you have, things that you think are interesting looking, surprises? Yeah.
So the biggest thing that I'm curious about is like octopi, right? Because they have a lot of structures and features that seem similar to things that maybe vertebrates have.
Yeah.
Like they have a beak,
Octopus have a beak. What else?
they have like internal organs
Yep.
that don't seem to.
They've got eyeballs. And also that octopus is representing all of mollusks. So I could have put a slug and you guys all would have been like, fine. But octopus is a mollusk.
Yeah.
A clam, I could have put a scallop up there instead of the octopus. So that mollusca clade is crazy. There's a lot of biodiversity. And you're picking up on something really awesome. This animal and this animal and this animal all have like certain things going for them that are absolutely independently evolved, which is really crazy to talk about. Okay, what else? How about where we go? You're the flamingo, by the way. How about where we go? There's always 1 or 2 people that are like, I didn't know starfish were so close to us. Starfish and sea urchins and sand dollars and sea cucumbers, those are all echinoderms. Echinoderms are extremely close to chordates, which is our clade, chordata. Our phylum is chordata. That's wild, I think.
What's the closest thing to vertebrates on there?
This thing? Oh, don't worry, you're gonna see it too much in a second. All right, just a little bit of animal diversity for you guys to chew on for a second. Those are the clades that are relevant for vertebrates. The other ones are just there for you to be like, oh wow, I gotta go Wikipedia octopuses. Good luck to you. Here are some clades, some synaphomorphies for these different clades that you are absolutely responsible for. These are things I want you to know. So there are many features here. The only synaphomorphy, the only shared derived character that I want you to know for Animalia is this is one of those times it's happened, multicellularity. Animals can be defined by multicellularity. That multicellularity in a jellyfish and you is the same multicellularity inherited from the common ancestor we had down here. So multicellularity for Animalia. As we go up the tree, things like jellies, which we haven't talked about a lot, but things like jellyfish, you guys know what they look like, right? They go in the ocean like this. They don't have eyes, they don't have a brain. They float on the currents, they have stinging tentacles when you go to the beach. Jellies are really important animals, they're very ancient animals. There's 2 features that jellies plus everybody else had. Sponges don't have this. So this is within Animalia. This node on the tree's got 2 characters. First is called diploblasty. And so when these animals, including you, are an embryo, there's an inside and an outside cell layer, tissue layer to the embryo. An ectoderm, ecto is the outside, derm is skin. So an ectodermic layer and an endoderm layer. Endo is inside, derm again is skin. The endoderm is the embryological origin of your gut tract. So from your mouth to the back of your digestive tract is all lined with endoderm cells. So 2 cell types. Also, things like jellies, and there's a couple kinds of jellies you can see on this tree, have something like neurons, synapomorphic with your neurons, to control their muscle cells, complicated movement. A sponge, individual cells go like this and water flows through a sponge. Jellies go like this. And they can be big. They have muscular control. There is not a central nervous system in a jellyfish. It's all dispersed, but there are neurons. You, of course, and so does that octopus, have neurons. As we go up the tree, there's a ton of characters that characterize bilateria. Bilateria is these 2 clades put together, right? Common ancestor and all the descendants. Bilateria is absolutely the worm clade. If you're a bilaterian, you're a worm unless you got fancy in your past. That's how you guys should think about it. All of these animals are worms or ancestrally look kind of wormy. And that's where most vertebrate phyla are. The features I care that you know about for bilaterians are, this one shouldn't be too hard, they are bilaterally symmetrical. If you cut them in half like this, there's mirror images on each side, or at least usually. And then part of being bilateral is this long word here, encephalization. It means you have a head. All your sensory organs are oriented on the front part of your body, and therefore you have a front and a back and 2 sides. You're bilaterally symmetrical, eyes, nose. Bilaterians add a feature that they shared with these old jellyfish here. We are actually triploblast. So when you were a little embryo, or if you've ever had a child, your child's little embryo has an ectoderm that's homologous with a jellyfish and an endoderm, but there's another tissue layer in the developing embryo, but it's still just a ball of cells that's called the mesoderm. So meso is in the middle, ecto on the outside, meso in the middle, endo on the inside. Mesoderm is where that cell layer, that tissue layer is where a lot of our organs and interesting bits come from. We're going to talk a lot more about mesoderm, probably actually in lab. That mesoderm has within it an internal body cavity. So you're not just like a tube with a food tube like a straw. You have between the outside wall of your body and your digestive tract a cavity, which is called a coelom. That word is pronounced coelom, and that's where a lot of your organs actually are. You have a digestive tract, which is sort of part of that same structure. And then really importantly, all these animals have, at least rudimentarily, a central nervous system. So it doesn't necessarily mean they have the thing that you think of as a brain, but they have a cord of neurons that goes through their body in some way and spreads out and helps the body function. And there's usually a control unit of some kind, even if it's just a swelling, a ganglia of nerves, not necessarily a brain, but a central nervous system. So at the deepest homologous level, Andy, like that spider has that central nervous system, has the triploblasty, has the bilateral symmetry, that, and the idea of all the sensory organs as a head, even if they're differently evolved, that is shared with you. So the octopus and the damselfly and the flamingo, these deep characters are actually homologous, even though they're of course very specialized in these different clades, which is really awesome. I want to show you guys a little bit of this, so it's not just words. Here's what I'm talking about when I talk about a coelom and these different tissue layers. So if we take a very simple bilaterian, that there is an earthworm, you can see that there's kind of these very basic divisions. That outer layer is coming from the ectoderm, the blue, the 3 tissue types, an earthworm and you are both bilaterians. You both have triploblasty. There's an endoderm-derived digestive tract in the very middle, and then between the ectoderm and the endoderm, there is a mesoderm, and that mesoderm has cavities in it that are called coelom on this side, coelom on that side, and those where organs are held between the food passage and the outside. Hope that makes sense. Here's what I mean by bilateral symmetry. I've done this a couple times. This is what I mean when I do that. Other animals, like a sponge or a coral, have symmetry, but they have radial symmetry. You can cut them like a pizza, and they'll kind of have these same functional units. Bilaterians are bilaterally symmetrical. You cut them in half long ways. That vertebrate, an owl, that arthropod, a lobster, are bilaterally symmetrical. So they're still animals, just like the sponge and the coral, but different kinds of symmetry. This is something that I think is really cool, and we'll talk about in lab. All bilaterians have that central nervous system where neurons are bound together to control different parts of the body. Almost all animals have a ventral nerve cord. You guys might remember from intro bio, if you cut up a worm or something, there's a ventral nerve cord, a tummy side nerve cord. Vertebrates famously have a backbone, and it is a dorsal cord. It's on the back of your body. What's really cool is that we can see in deep animal evolution, in developmental biology, there was a mutation at one point, and we flipped. So we're the weird ones to have the cord going down the back. But these deeper structures are very much homologous. That's really wild. The thing that's fun about bilaterians, and I know this is a gross picture, but I think it's hilarious. This is a leech eating an earthworm. So this is an annelid eating another annelid. The most basic biology of bilaterians, even if you're talking about something like a bird, you can boil it down in certain aspects to call it, it's just a tube within a tube. Then we have sensory structures and locomotion and reproduction and all kinds of cool stuff that gets added on. But you can think about it, all bilaterians is basically tubes within tubes that live. And why I like this picture, it made me laugh when I first saw it, because that's a tube within a tube within a tube within a tube. Because when that leech eats that worm and swallows it whole, you got four tube layers in there. Okay, anyway. All right, looking at our time here, I want to give you a little bit more just animal evolution context that I think is cool. So this is you're gonna see a couple of these in this class, but really not too many. This is a time calibrated phylogeny. So it's the same evolutionary information you've already seen. The connection between the groups is the same, but down here is actual geological time. So the Ediacaran period, the Cambrian period. These white arrows represent actual fossils. We have fossils of annelids that go back this far, fossils of mollusks that go back this far. Here's you. This bass is representing us chordates. That's our phylum right there. That's why it's capitalized. So this is our actual like geological record of animal evolution. And then the evolutionary relationships are overlaid on the timeline. This whole thing allows different kinds of scientists to have different ideas and to test hypotheses. So I just think it's cool for you guys to see these relationships and then this geological aspect. So go ahead and just chat with each other for a second. Any observations you can make or anything that makes you be like, oh, I wonder about this. And then how could we maybe test the thing that you're wondering? So let's take a few minutes on this, take this in again. Again, same evolutionary information, but with a time component. Go ahead.
It also looks like shoulder top. How do you think?
One of the shop should be lost.
Yeah.
There's your friends.
All right, what are some observations people have? This is basically the state of human knowledge right now. Yeah, that's it.
It looks like Bilateria existed for quite a long time before it like split up, but when it did split up, it split up like all at once.
Amazing. What do we think about that? Bilateria. So you can take the genes, you can take the genetics of all these animals and get an idea of, use a mutation rate, do some math and get an estimate for when, based on their DNA today, they probably diverged. And that divergence is always way back here. But somebody like me who like looks at rocks all day is like, well, the fossils only go to here. That's extremely interesting. What other things people notice or maybe have a question about?
So happens in the early mid-Cambrian where.
Anybody heard that word Cambrian before? In what context? Like the explosion. It's called the Cambrian explosion. It's this phenomenon that everybody knows about. Basically, the fossil record starts here because all these different animal groups start evolving hard parts. They're suddenly shells and kind of a bony kind of thing. And that's what actually makes fossils. Isn't it interesting that this animal, which has this thing on its back, yes, a longer fossil record for mollusks than for these smushies, not hard until this time. So the fact that mollusks have shells probably shows us that they have a more ancient record, but that might not be because they're actually older. It might be because their bodies fossilized better earlier than the other clades. Isn't that freaky and upsetting? The things we have to worry about. So, okay, that's cool. That's what I want you guys to take in and just think about. It's very cool and important to me that we don't just start with the fish and you get that context. So here is our clade of deuterostomes. So this node on the tree, this is a clade that we chordates, we vertebrates are a part of. This is called deuterostomia. It's always simplified as being like us and starfishes, but there's actually several phyla that were on that first slide. This is a phylum, this is a phylum, that's a phylum, that's a phylum. Oh, sorry. This is phylum, phylum, phylum. There's 3 of them on here. Doesn't matter what they are. With some of the clade names labeled, only the ones that are all capitals actually pertain to us vertebrates, and you can see we're actually almost there, so I'm excited. These are our closest relatives that aren't themselves vertebrates. Things called tunicates, that weird pink thing you asked about a couple slides ago. This thing's called an acorn worm, and then there's all the echinoderms, which are things like sea urchins and starfish. Those are our closest relatives amongst all the other animals. Isn't that upsetting? How about that tunicate right there next to us? Kidding me?
Are there any living examples of the two?
Every single one of the things on this slide is a living organism. There are hundreds and hundreds of kinds of tunicates today. I'm going to show you some, don't worry. The only feature, well, there's 2 features of deuterostomes that unite all these different groups of animals. And the first one, is not going to be hard for you to remember, is deuterostomy. I'm going to show you what that is here in a second. The other feature is that at some point in their development, some of them only when they're babies, if you haven't seen what a baby starfish looks like, do some Googling, have pharyngeal gill slits. Now, what does that mean? What are these anatomical features? They both come from developmental biology. So even 120 years ago, scientists could look at embryos developing, and people put things like starfish with animals like us because our embryos develop in the exact same way and completely unlike other animal embryos. That's really, really cool. So developmental biology is a field we're going to come into again and again, where you're looking at the development, usually of very young, going from like sperm and egg cell to like the born animal, where we can infer a lot of evolution and then test new ideas. So here's what deuterostomy means. If you remember, the 2 kinds of bilaterians are deuterostomes and protostomes. Proto is like first, deutero is second, and stome is mouth. So second mouth and first mouth. When there is a fertilized egg that is becoming an embryo and divides into 2 cells and then 4 cells and then 8 cells and then 16, and becomes a little thing. There's a moment when the cell starts to cleave and the animal starts to form. There's always a first hole that forms in that ball of cells. That hole is called the blastopore. And in protostomes, the mouth first clade, so something like a crab or an arthropod, something like an octopus, that first hole eventually becomes the mouth, and a hole that forms later is the anus. And so there's a coelom in the middle, mesoderm, endoderm, ectoderm. But the thing here is that the first hole becomes the mouth, the second hole becomes the anus. In deuterostomes, which is definitely you and starfish and tunicates, that first hole that forms actually becomes the anus, and the second hole that forms is the one that becomes the mouth. So in our clade, the digestive tract kind of goes in reverse in terms of how it forms. That is probably just some dumb fluke of developmental biology that happened one time and stuck, but it's something that unites all of us and we can recognize in these other animals. And so it's really cool. Deuterostomy, we've known about it since like the mid 1800s. And that is why we go with these animals. Interesting. The other feature that all of these deuterostomes share, at least at some point in their biology, are these things called pharyngeal gill slits. So a pharynx is like your throat. That's what you're going to hear that word talked about a lot. These animals have very different anatomy than us, so there's not a lot of throat actually happening. But water comes in to the throat and then comes out through gill slits. There's slits in the pharynx that water passes through, and in some of these animals, there are gill structures that take oxygen from the water. So a gill structure thing, the important part here though is not all the details of the gill structure, it's the actual slits themselves, unite all deuterostomes. This is something that's really, 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 really incredible to me and something that people have known about for a long time. This is like a human fetus at relatively early development. There's the little eyeball, here's going to be the little backbone. And there's a moment in human embryonic development where we have these gill arches. If you were to follow a shark, those things become the actual gills. In you though, of course, you don't have gills, but in your developmental biology, those structures, and then the important thing for this slide are the slits between them, become all these different structures. This is bone, cartilage, this is muscles, this is nerves. And you can see how nerves, muscles, and bone all together as units in your face come from the different gill arches that in a shark are just like pretty boring, but in you look crazy. There's a cool GIF I want to show you guys just to give you a little bit of context for the bigger oh, screen. Yeah. Okay. I just really like this because I want you to look at this for a while. This is a human embryo and a dolphin embryo, both just being shown as they develop next to each other. Take it in for a second, because this is how developmental biology can help us understand evolutionary change over time. So make some observations here about that dolphin, about that person. These are obviously visualizations. We can take fancy CT scans of dolphins in aquariums, and ladies when they go to the doctor, and get these visualizations. So, okay. Some people say gross. That's fine. We noticing anything? The thing that I care that you notice is that this dolphin and this person both at different times have those pharyngeal gill structures with the slits between them. Then of course they go away. I know it's upsetting, but it's also very, very awesome. Cass couldn't help. For a hot second, the dolphin gets a little femur and then the femur goes away. What? We'll talk about it later because we're definitely not there yet. But I just want you to see the pharyngeal gill slits in a person. And this is what I mean when I talk about developmental biology. We look at the development of embryos. Okay. We are almost there today. Okay, so here's our geological time scale again. Oh, I know. I was just going to show you guys that animal diversity kind of moment because all these plates show up at about the same time. So on this geological time scale, early in this period called the Cambrian, which is the very start of this new eon, the whole reason this thing is even called something different than this thing is because this is basically where fossilization starts. You get good bones, good shell. Later, you get plants that make wood. Hard parts start in the Cambrian. The Cambrian is kind of famous. You guys maybe have heard about it. I'm not going to go into it far here, but there's tons of insane animal diversity in the Cambrian. Some of it is related to the phyla we have today. Some of it is totally not at all. Some of these things have 5 eyes and like a claw mouth on a tube. We're not going to talk about them because they're not vertebrates, but I just wanted you to see and understand how cool the Cambrian is. In the Cambrian, though, there are chordates. There are members of our phylum. If you already know, don't say anything, but do we see any chordates on this nice Canadian painting of the Burgess Shale, which is a fossil site in Canada that preserves the Cambrian? Where's the chordate? Where's your friend, your closest relative? It's not this one, much as you might want it to be.
Yeah.
What do we got? What are we saying? You were pointing. What are you pointing at?
Most on the right, the biggest one on the right.
This one? Biggest one on the right, I'm sorry. This one?
Yeah, that one.
Oh, that one. That's the same as this one. Those are arthropods related to like spiders and bugs and stuff. Not a bad guess.
This is top left.
Top left, that's us. Pikaia is its name. That's one of the earliest chordates that we know about. So a member of our phylum Chordata. There are actually several chordates known from the Permian, from the Cambrian at this point. So there's Pikaia. This is what it looked like. Here's a nice fossil of it from Canada. So there's its little face, little antennae. This is a fossil from China, haiku ichthyes. Oh, the haiku fish, isn't that nice? And then this one, not as fun, Metaspergina. Both haiku ichthyes and Metaspergina, these are all Cambrian chordates, but these two, haiku ichthyes, Metaspergina, are actually truly vertebrates. They're part of Vertebrata. They have a lot of features like actual eyeballs that only vertebrates have. So Pikaia is a chordate just like us, but there's also Cambrian animals that represent our actual clade we're in called Vertebrata. It's really exciting. So there are, and we can probably do this, 4 characters I want you to know that unite all chordates. So vertebrates are one kind of chordate. These are also chordates. There's a notochord. A notochord, do we have time? What time is it?
It is.
Oh, 12:14, we can, okay. So a notochord, you can at least write it down. I'll say it again on Tuesday. Notochord is a stiff rod that does not bend, collagenous or made of collagen, and like the most primitive animals. It's the rod that adds structure to the internal body. You can flex your muscles against the notochord, and that's how a fish goes and swims through the water. So a notochord, a tough, non-compressed cord. Not surprisingly, laying on top of that hard structure is that actual nerve cord. So some sort of control unit that runs the length of the animal. I bet this is familiar to you. A rigid thing and then the actual spiny kind of thing laying right on top of it. So that dorsal hollow nerve cord. It's another chordate feature. There's also a post-anal tail. So another way to say that is a caudal fin. So what that means is post is after, anus is anus, and then tail, a fin on the back of the animal. A tail fin is what we call it in almost all fishes and animals today that have it, a tail fin or a caudal fin. So this post-anal tail, the notochord, and the dorsal nerve cord. Actually, I think there's only 3, and I messed up. There's 3 characters I want you to know that unite all 4 dates. And I don't think we're going to get to it yet. I'll save that for Tuesday. We're almost there. Gosh, I thought we'd make it to vertebrates. We're so close. All right, see you guys.
Thank you, Dr. Pico.